So, hello everyone, my name is João. I am a PhD student at University of São Paulo, Brazil, and also a visiting student at Professor Enderman Lab here in University of Wisconsin Madison. And today I'm going to present my work. It's Genome-Wide Association for Drought Tolerance Potato. It's a partnership with International Potato Center. So, oops. Wait, okay. So in this report published in Science last year, research alerts the fact that children born nowadays may experience 6 times more extreme heat waves compared to their grandfathers. And these extreme events like crop failure and drought seasons can impact put food security in risk, especially in developing countries. And potatoes is a major crop for these regions. And so extreme conditions requires varieties adapted to these conditions, right? And heat and drought are two of the abiotic threats that we must face. Compared to other crops, potatoes are not too heat tolerant. We can see in this graph what happened with dry matter when we move from cold conditions to hot conditions. There is like a loss of dry matter. And also we know that potatoes are a very water efficient crop, but drought seasons can impact severely potatoes, especially in some stages like emergency and tuberization states. So SIP have 4 major target environments. In each target environment, we have different target traits. And we can see in this table that LTVR population is present in 3 of the 4 major target environments. So LTVR stands for low-land tropics virus resistant, is a population that started in the 70s. And the end targets nowadays are earliness, heat and drought tolerance, and processing traits. And in order to understand better the genetic response for drought tolerance, we carry out a GWAS analysis to understand better what's happening in the population in cycle 7. So 2 field trials were performed, one in La Molina, another in Majes. Both environments are classified as semi-arid according to Köppen climate classification. Also, they are in lowlands with a range of temperatures, as we can see, considered high for growing potatoes. In both field trials, all the clones were submitted to 2 different treatments, the drought and the full irrigation. In the beginning, the same amount of water was applying in both treatments, and the deficit started when 5% of the plants had flowers, a morphological sign of tuberization, and the terms of irrigation depends on the management of the water in the soil, but on average 2 or 3 days for the full irrigation and 15 days for the deficit treatment or the drought treatment. The traits evaluated were wilt, tuber set, and dry matter. And 655 clones were genotype and phenotype. The clones were genotyped with target amplicon sequence that Professor Enderman talked earlier, and that generated 2,244 markers after quality control. The dosage call was done using the UpDog package, and all the statistical analysis was done using the stage-wise pack that Professor Enderman developed. We had different experimental designs for each trial, so it fits very well to our data. Here we can see the impact on drought and on wilt. There is a decrease of 62% from one level to one treatment to another. And unfortunately, we didn't find any super. But we can see there is a distribution of loss that we have to work to find more stable clones to understand, to get clones improved for drought tolerance. Also, the same behavior for tuberset. We can see there is a decrease of 47%. And to dry matter, we don't see the same behavior. This is because it looks like that some clones gained some dry matter, but it's more likely that these clones lost water than rather than actually gain increased dry matter from one treatment to another. Here are the heritabilities in the plot level. We can see that lamolina in general had more higher heritability values, and always we can see a decrease from the fertigation to the drought treatment. And this is because we have a bigger residual effect. We have the treatment, the drought have a bigger impact on the phenotype of the clones. One important thing to look is the yield in both treatments, and when we plot the yield in function of the maturity group or the senescence group or score, sorry, we can see in the left side the yield in fertigation there is no pattern, but in the right side we can see that yield in the drought there is a linear trend that early clones yield better compared to late clones. And maybe there is a relationship. We can see this in a genome, in the genotypic data. And we can see there is some relation. In the left side, we cannot see any association for the yield in fertigation, but there is a peak in the right side. There is the drought in the yield in the drought treatment. And this peak in the beginning of the chromosome 5 is near of the CDF1 gene. There is the same gene the same peak that we found for senescence in the left side. We can see it's the same marker. So CDF1 is known to be responsible for the indirect induction of tuberization, and makes sense to be significant for our population. The stress is applied in the beginning of tuberization, so early clones can escape more easily from the drought impact. Also, in this paper, the authors point out about a different response to the abscisic gas. Plants with the alternative allele responds to ABA with lower stomata aperture compared to the reference allele. Also, they talk about the regulation of stomata size and number on the leaves, and all these factors together leads to a lower water loss. But also in this paper, they point out about the reduction of thickness in fertigation. And we don't see the same effect because in the Bahata plot, we cannot see this in the results from that Professor Enderman showed earlier. We cannot see the same results. So one explanation of we don't see this fitness reduction is because our trials were performed in heat environments, and CDF1 is also related to heat tolerance. So as conclusions, CDF1 appears to be important for drought tolerance. We have to dig it out more, but it seems like it's an important gene for this trait. Also understand the dynamics of the CDF1 in the population level. It's important to breeding purpose. I would like to acknowledge International Potato Center, especially to Elisa Salas and Mokhtar Kant about all the support and the data for my university from São Paulo, University of São Paulo, my lab colleagues, my advisor, Fernando, also Professor Enderman and all my colleagues from the lab here in Wisconsin. And thank you, David, and all the organizers of Organization of Institutes of Polyploids for opening this place. And thank you very much for everything.